correlative study is designed to examine the part of the hypothesis focusing on firstly the relation between the number of ticks and habitats providing food for hosts, and secondly the vegetation cover in relation to the number of ticks. To do so, three types of food availability habitats were determined: a high food availability habitat, an intermediate, and a low food availability habitat. In the research, we did not measure food availability directly because of practical reasons. Instead, the main tree species of each stand is used as an indicator for food availability. Based on literature, Vagus sylvatica and Quercus robur stands are classified as high food providing habitats for mice, Pinus sylvestris and Pinus nigra as intermediate habitats. Pseudotsuga menziesi and Larix campheri stands are classified as low food providing habitats. The correlative study is carried out in the Oosterang forest. Based on the stand map of the Oosterang forest, the different food availability habitat types are identified. From the identified stands, a random sample of 60 stands was taken. So for each of the three food availability habitat types, 20 stands were taken. The sampled stands were at least 200 meters apart from each other to ensure independency. Within each stand, the number of ticks were collected on a randomly chosen transect of 25 meters. Ticks were collected with a 1 meter wide cloth sheet dragged on a steady pace over the vegetation. Together with collecting ticks, other variables were measured. Firstly, undergrowth vegetation cover was measured to be able to examine the relation between cover and tick abundance. Vegetation cover was measured each 5 meters of the transect for 3 strata. These 3 strata were 0 to 15 centimeters, 15 to 50 centimeters, and 50 to 150 centimeters. This was done to account for cover occurring at different heights. The cover for each stratum was averaged for every transect. Canopy cover was measured on 0, 5, 10, 15, and 20 meters using a circular tube. At the very same points, vegetation height was measured in centimeters. Both canopy cover and vegetation height were averaged for each transect. Lastly, the dominant undergrowth species was determined. The whole procedure resulted in a number of ticks, an average vegetation cover per stratum, an average canopy cover, an average height, and the dominant undergrowth species per sampled stand. The standard deviations for the averaged variables like canopy cover and undergrowth height were used as a variable indicating the heterogeneity, since it provides information on the variation within each averaged variable. The rationale used within our conceptual model leads to the prediction that the number of nymphs will be significantly higher in habitats containing food-providing trees for small mammal hosts. To analyze the relation between the number of counted nymphs and the food potential habitat for small mammals, a generalized linear model is used. In this model, the counted number of nymphs is the dependent vector. Host food potential habitat type will be the nominal fixed vector, containing 3 levels: high, intermediate, and low. To account for the additional variation, covariables will be used. Since we expect a relation between the undergrowth vegetation cover and undergrowth vegetation height, these are included as covariables. Canopy cover will be included since literature highlighted the importance of microclimate for ticks. Covariables are all continuous variables. The model will be run using a backward selection method, in which all non-significant variables are excluded. Due to the fact that the number of nymphs consists of count data, the model needed to be run using a Poisson distribution. It appeared that the high food potential habitat and the intermediate food potential habitat significantly differed from the low food potential habitat. All covariables turned out to be non-significant. The final model therefore consisted of only the habitat type. The prediction that high potential food habitats for mice will be the class containing the most nymphs is therefore not met. However, the low food availability class contains significantly less nymphs. Furthermore, we assume a linear relation between the undergrowth vegetation cover and the number of nymphs, as well as between undergrowth vegetation height and the number of nymphs. These relations will differ for the different food potential habitats, resulting in higher intercepts for the better food potential habitats. Again, a generalized linear model is used to analyze this prediction. The independent variables, cover and height, are included as covariables. The explaining variables are singly entered into the model. The variable accumulated undergrowth was added to describe the cover over all 3 layers and consists of the sum of these layers. The scatter plot represents in all cases the number of nymphs on the y-axis and the independent variables, different covers, height, on the x-axis. When analyzing the graphs, it becomes clear that all variables show weak relations with the number of nymphs, which is reflected in the low r-squared values. Furthermore, it is interesting that most relations are negative, which implies a decrease in the number of nymphs with an increase in value of cover or height. No significant relation was found for cover and height. Apparently, undergrowth vegetation cover and undergrowth height are weak explanatory variables for nymph abundance. An essential unanswered question within our research remains about the causal mechanism that can explain the significant difference between high and intermediate food potential habitats and low food potential habitats. In order to examine this question, on the basis of our dataset, a canonical redundancy analysis or RDA was carried out. In this analysis, the gradients within tick abundance, this means the abundance of adults, nymphs and larvae, are examined for the different plots. The undergrowth covers, undergrowth height and canopy cover are used as an explanatory environmental gradient. Furthermore, dominant undergrowth species and stand species are used as nominal environmental gradient variables. This generated a species environmental canonical analysis. The number of ticks were log transformed in order to reduce the large variation. Here you can see the ordination diagram that is displayed as a result of the canonical RDA analysis. The axes represent the length of the gradients as calculated by the canonical analysis. The plots are represented as the different dots. Plots with an A refer to the high food potential habitats, B to the intermediate and C to the plots with the lowest food potential habitats. Plots for the different food potential habitats are not very strongly clustered. However, some patterns can be observed. More importantly, there seems to appear no strong explanatory gradient of undergrowth species, cover, height, canopy cover or stand type. In the statistical output, it becomes clear that the explained fraction remains relatively low, since only 0.22 is explained by our measured gradients.